Hello, everyone. Welcome back to the TBSCNW workshop for the second day of our workshop. Today, in the morning session, we will have two speakers. So I'm going to announce now the first speaker, Domenica Veniero. It's a great pleasure for me to announce her presentation, as she is a very good friend and colleague. So Domenica started her career at the University of Brescia, where she obtained her PhD. Then she traveled and worked at different institutions, like the IRCS Santa Lucia and the University of Glasgow. She's now assistant professor at the School of Psychology, University of Nottingham. Her research interests focus on the role of brain oscillations, both in long-range connections and also for local activity. And also she's interested in studying the effects of stimulation on cortical excitability and behavioral performance. So please welcome Domenica Veniero for her talk.
Okay, can you hear me? And can you see me? Yes. So good morning, everyone. Thank you, Marta. It was very nice. Thanks for having me today. I just want to say I'm so sorry because there's going to be sneezing and there's going to be some coughing during the presentation, so sorry. Okay. So as we saw yesterday, there has been, for the past 10 years at least, a slightly a small change, let's say, in focus of what we are doing as neuroscientists. So in the past, if you think about research, what we were interested in doing was trying to understand the brain-to-behavior relationship, in the sense that we wanted to know which area was mainly involved in one specific function. So I don't know how to use it. For example, in this case, we have 3 areas, and maybe we wanted to know which of these areas was involved in attention or language, so on and forth. But now, with the recent development in neuroscience, what we are starting to understand is that there is no area that is responsible for one function. But what we want to know, rather, is which networks or ensemble of areas work together to, in order to help a specific brain function. So we are not interested anymore in these 3 areas alone, but rather we want to map a network, a functional network. So we're not interested in these 3 areas, but we are interested in understanding how these 3 areas, for example, work together. And their functioning together and orchestration of their activity is able to explain how we perform tasks or any function, really. So the main idea and the main, I would say, topic of my talk is going to be summarized by the first sentence on this slide. So cognition, anything about cognition, so from basic perception to very complex functions, requires the integration of information that is processed by different areas. And we can look at connectivity, of course, in different ways. And maybe the first thing that comes to mind, of course, is anatomical connectivity, which we think is relatively fixed over time. But at the same time, there must be some kind of mechanism that is faster and that is flexible, that can allow different areas to talk to each other, and that these talking to each other will subtend specific function. So this mechanism that I will talk about in a minute must have some characteristics. So there are some criteria. The first is that this must be an efficient mechanism. So anything that gets put together in terms of network must be appropriate to the goal of, that we have at any specific moment. It also must be something that is fast, because of course we do several things, and maybe the demands from the environment will change very quickly, so this mechanism must also change very quickly. And the other thing is that this mechanism must be flexible. And what I mean by flexible is that areas that are working together have to quickly stop working together so the other areas can start communicating together and working together. So flexibility means that areas that are linked at some point might not be linked anymore, and this being linked and not being linked must happen very quickly. So I guess my main point, or the main point of what I'm doing, is that it's important to know what is connected, but what I'm interested in is also how different areas are connected. And most importantly, what I've tried to do is trying to understand how the communications between different areas can also shape our performance and therefore our behavior. So one mechanism that has been proposed over and over that should explain or should have this characteristic of being flexible, fast, and efficient is brain oscillation. Because they are quite fast and they seem to respond to these three characteristics very well, so they meet these criteria. So I don't know if you're all familiar with brain oscillation, but these are patterns of activities that are usually categorized according to their frequency. So these are like a very classic picture of what we know about oscillation. So they're usually categorized according to how fast or slow they are. So we have delta, theta, alpha, beta, and gamma, and each one of them is characterized by a specific frequency, and in particular, my favorite is beta, which is around 20 hertz. So there is a lot of research about how much each of these frequency bands contributes to each function, but something that I'm really interested in at the moment is also another index, which is the phase. So it doesn't matter which oscillation you're looking at, so beta, alpha, or whatever you're interested in. There's also another index that seems to be pretty interesting, which is the phase. So the phase is simply where the oscillation is at any specific moment in time. And the interesting thing that has been suggested and sometimes proven, some other times not so much, is that there are phases that are also referred to as good phases. For example, let's take this peak here. What happens is that if we take as an example the visual cortex, when the oscillation is at this stage, so the good phase, there's a certain number of spikes. And if we present someone with a stimulus at this good phase, the participants are tendentially going to say that, yes, they have perceived the stimulus that we are presenting. However, there's also something else, which is this phase here, which we usually call the bad phase. If we look at spikes, tendentially, let's say usually, the number of spikes here is reduced. And if we present exactly the same visual probe as before, participants will tend to say that they haven't perceived anything, so there is no stimulus. So because there is this relationship between spikes and also performance, visual performance in this case, the idea is that the phase of an oscillation will reflect the excitability of the cortex that is generating that oscillation. Okay, so the question that I will try to answer during the talk is it possible that this phase, so this index of excitability, is somehow mediating communication between areas? So, and in particular, my studies, I've tried to see whether a reorganization of phase is actually able to explain how different areas can communicate. This is just a very brief summary of what to expect. I have two studies that I'm going to show you that are about visual spatial attention, so the visual system, and then briefly I will show you a study that deals with the motor cortex, so the motor system. Before we go on, I just wanted to have a note about the methodology that is often used in these studies that I'm going to show you, that is brain stimulation, so TMS, together with EEG. So we've seen yesterday as well that usually what we do is we want to apply TMS, or brain stimulation, as a mapping tool. And the idea is that we apply TMS, maybe repetitive TMS, And in this case, we want to have what was called at the time virtual lesion. So we want to interfere with the activity of the area, and then if we get any change in behavior, then we tend to say that that area is responsible causally for that function. And for this reason, we tend to associate the word causal with the brain stimulation, with TMS in particular. However, in the studies that I'm going to show you, I've used TMS in a slightly different way. So the idea here, also we saw yesterday actually, is that when you stimulate an area, again, for example, the area here that it's in the prefrontal cortex, what happens is that the TMS will actually activate the area that we are stimulating, but also all the connections of that area will also be activated. And this means that we can trace the connectivity of that area, because the activity, as I just said, that starts here in this area will spread towards interconnected area. Of course, if we just use TMS, there's no way of telling what's happening in other areas, because usually we use TMS on one target, maybe two areas, so we have no way of telling whether or where the activation induced here is ending up. So what we do in this case is to, sorry, I went too fast, but what I've done so far is basically using EEG to try and trace what happens in other areas once we stimulate a node or a target area. So the interesting thing when we use TMS and EEG together is that we have a massive advantage, because as you can see here in this picture, what happens is that by design, we know which area is starting to communicate with other areas. So here I call this area that is stimulated by TMS the sending area, because we know we are stimulating that area. And any other area that is receiving the activation is, of course, the receiving area. So the nice thing is that by design, we know the direction of this communication, and therefore we have information about effective connectivity. We don't have to apply any model to that, because we already have this information. So as I said, on the other hand, we have this other technique, which is EEG, that has the advantage of recording from everywhere, let's say on the cortex or on the scalp, but of course has no information about causality in itself. So the idea is to put together the causality that comes from TMS and the global information that is coming from the EEG. So our task with EEG is just to check what's happening in other areas that we are not stimulating, but that we think are associated with the stimulated area. Okay, so the first study that I am going to talk about is a study about connectivity within the circuit that is usually associated with spatial attention. So this is just to introduce spatial attention in a funny way. So this is the main square in Nottingham, and at the moment there is a nice Christmas market. So imagine that you arrive at the Christmas market in Nottingham and you're looking for your friends. It's going to be quite challenging because it's very complicated. But what you will do very likely is you will start to scan your visual scene, and at any given moment you will attend a specific location of your visual scene, and this means that you will enhance any information that is coming from the attended area of the visual scene, and you will tend to suppress the information that is coming from the rest of the space. So that's the job of visual spatial attention, is to enhance the neural processing of the stimuli that are falling within the attended location at the expenses of anything else. Okay, so we started by thinking, okay, what are the areas that are associated with visual spatial attention? This is nothing new. Of course, the frontal fields are always associated with attention. And we know for sure from a series of studies in animal and humans that frontal fields are able to send top-down signals to visual areas. So the idea was, if we keep in mind the definition that we have of phase, where phase is an index of excitability, the question was, is it possible that when we allocate attention in space, what happens is that the frontal fields control somehow the excitability of the visual cortex by shifting the phase of the oscillations that are generated within the visual cortex. And if you think about it, this is a nice way of deciding, in a way, which stimuli will be processed, because they will fall in the good phase, and which stimuli will not be processed or will not be optimally processed because they fall in the bad phase. And another advantage of moving or working on this excitability phase is also that the frontal fields can align their own good phase with the good phase of the visual cortex, and this means that there is a very good exchange of information between the two areas, for example. So the main aim of this study was to test how these frontal fields can control the visual areas. And in particular, the hypothesis is that the frontal fields send top-down signals to the visual cortex and generate what we call a phase reset of the activity of that area. So this is a visual summary of the hypothesis. Let's just look at this for the moment. So we are activating the frontal fields. What we expect is that the frontal field will send a message to the visual cortex, because we know that they are connected. And this is the activity of the visual cortex. So the line here is the moment when the signal from the frontal fields is reaching the visual cortex. And the phase reset is what happened afterwards. So as you can see here, the activity of the visual cortex is kind of random before, but then after this top-down signal is reaching the visual cortex, there is an alignment. In order to, for this to happen, of course, it also means that there is some kind of reorganization of the phase. So now the activity is nicely aligned. And this part is what I said, but I'm not going to test this specific hypothesis in these studies. It's just to show you that if this is the case, then the frontal fields can align its own good phase with the good phase of the visual cortex. So as a consequence of this phase reset and this nice alignment, it also means that if we look or if we test the excitability of the visual cortex, no matter what we use to test this excitability, we should always be able to trace this up and down in its excitability, and that's what we have done. So you will see this picture quite a lot in the rest of my presentation, but it will become more and more clear why. So when we started to plan this study, we were inspired by a series of studies in monkeys. They're very interesting because what they show is that when we stimulate the frontal fields with electric stimulation, so invasive stimulation, there is, of course, neuronal spikes, so there is a specific pattern. But what happens is that if you ask the monkey to do a task, and then at some point there is this voluntary shift of attention towards the target, the spike profile is very, very similar to the one that was triggered by the activation of the frontal fields by electrical stimulation. So we thought maybe we can do the same with TMS. So we can use TMS to emulate the generation of an attentional signal that eventually, or we were hoping, will reach the visual cortex. Yeah, okay, this is just to remind you that in this case, the idea is that the frontal fields are the sending area, and we were hoping to find something in the visual cortex. So we were hoping that the visual cortex is one of the receiving areas. The experiment actually is very simple. It's just a single pulse TMS over the frontal fields. The idea is to trace with the EEG what happens to other areas. So there's a single pulse, and then there's a sham stimulation, and participants were just asked to rest. And then all the results that I'm going to show you are just the difference between the real stimulation and sham stimulation. It's not particularly relevant, but maybe we might want to discuss this a bit. So the first result that I'm going to show you is about the EEG. And this index that is called intertrial phase coherence. And this is just a very nice way of checking how much at each trial, there is the same pattern in ups and downs. So how much alignment there is over time. And it's a very classic way of defining or checking that there is this phase reset. These are the results. What I'm showing you is just the average of a lot of channels. It's just a summary. So this is the intertrial phase coherence. The more, the redder, the better. So we were looking for quite red stuff. And 0 here represents the moment when we are delivering the TMS over the frontal fields. The square is everything that is significant. So you can see here that there is some kind of phase reset around 29 hertz, so high beta frequency. That seems to be over the electrodes underneath the coil. And then there is something around 15 hertz, so again, lower beta, that seems to be over the occipital electrodes. And then there's something around alpha, so 8, that is over the central left electrodes. So I think this is like super interesting because it seems to suggest that there are possibly different areas that might respond at different frequencies, but it's not something that we have investigated further, because at the end of the day, what we were interested in was the occipital electrodes. So this is exactly the same. I'm just showing only the occipital electrodes. So what you can see here, if you look at this side of the EEG, is that there is a nice phase reset as measured by the intertrial phase coherence around 15 hertz. That starts when we deliver the stimulation and seems to last for around 200 milliseconds. There you go. Okay, so let's say that this was an indication that there might be something there, that there might be some kind of phase reset going on. Then the next question would be, okay, even if there is this phase reset, is this relevant for our behavior? Is there something that happens when we are doing some kind of task? So we decided to, again, stimulate the frontal fields and try to measure what's the effect of activation of the frontal fields. So if you see here, participants were just doing something very simple, which is discrimination of movement, so dots moving left or right. Their task was to say whether they were moving to the left or the right. The only thing that was happening is that the frontal fields was activated, and then after some intervals, the moving dots would start. And we tested a lot of intervals from the frontal field activation. And the idea is, again, that if it's true that there is phase reset, so the excitability of the visual cortex is nicely aligned by these oscillations, what would happen is this. So if we test all these intervals, we should be able to reconstruct the ups and downs of the excitability of the visual cortex. These are the results of this experiment. You can see here on the y-axis, that's the hit rate, so how good participants were. On the x-axis, you can see the time from the activation of the frontal field, so from 0 to 200, and then we have a second window because we wanted to see even farther, and the second window goes up to almost 300 milliseconds. So what you can see here, I hope, is that participants' performance actually oscillates. So you can see that it goes up and down over time. And this is true soon after the activation of the frontal fields, but also in a later window. So this means that we are actually able to see that the excitability of the visual cortex is fluctuating over time, and that is somehow related to how we behave, so how good we are in discriminating the motion. If we look at the spectral content of the performance of these participants, which is what you can see here, we saw that it's in the beta frequency. So in the EEG experiment, there was this nice red blob around 15 hertz, which is beta. And still, we can find this beta in the behavioral performance. Sorry. Okay, so after these experiments, then we wanted to know if these changes in excitability that kind of shape the performance of our participants depend on the excitability of the visual cortex. So remember that at the beginning I said, okay, we always, no matter what we use, any measure that we use, we will always be able to trace these ups and downs if there is a phase reset. So we used a totally different measure this time. We stimulated the visual cortex and I think you know that if you stimulate the visual cortex with TMS, in most of the participants, you should be able to elicit these phosphines, which are visual illusions. The more phosphines participants can perceive, the more active and excitable the visual cortex should be. So we activated again our sending area, which is the frontal fields, and then we activated the visual cortex. We also had more questions, so we came up with this very complicated experiment that was very long, 6 hours, I think. So the first question was, okay, the frontal field seems to have an effect on the visual cortex, but on which visual cortex? So for this reason, we tested 2 connections. So the frontal fields and V5, and then the frontal fields at V1. The idea is, therefore, that we stimulate the area of interest, and then after different intervals, in this case 19, we stimulate the visual cortex. And then we also wanted to know whether there is any unspecific effect of TMS, so we also have a control condition, very classic control condition, where we change the frontal fields for the vertex, but the visual stimulation remains the same. So these are the results of this nice experiment. You can see here on the y-axis the number of phosphines reported by our participants as a function of the interval from the frontal fields activation. In particular, this is the stimulation of frontal fields followed by the stimulation of V5. And you can see that the number of reported phosphine is actually fluctuating over time quite nicely. And it's kind of persistent. It's going on for quite some time. And this is the control condition. So now this red line, which is a nice 17 hertz, which explains this performance, is the same that I'm plotting here, just to show you that it's not nicely explaining, or not so nicely explaining the number of phosphines when we replace the frontal fields with CZ. First thing that we wanted to know is whether there is a difference between control condition and real condition, and there is such a difference in the beta band. But this is just a check for us to make sure that we could continue with other analysis. These are the data from each participant. It's exactly the same. So you can see number of phosphines and the time from the frontal field activation. And I thought it was quite nice because you can see that every participant is kind of going up and down in the number of perceived phosphines. If we look at the spectral content of their performance, again, we can see that there is a nice beta band that explains the performance, so between 15 and 19 hertz. This is what happens in a later window. It's not so important, but the main point that I wanted to make here is that even if there is an effect on these phosphines perception, this is short-lasting. If we check for further window, we don't find any difference with the control condition. So let's see what happens when now we stimulate the frontal field. So we're interested in understanding what's happening on the visual cortex, the primary visual cortex, V1. You can see here again a number of perceived phosphines and the time from FEF activation. Again, this is what explained the performance for the V5 condition. It's just as a comparison. This is what happens for the control condition, and unfortunately there was no difference with the control condition, so there was no point really in going on any further. Yeah, this is just to show you that it's true in the first window, but it's also true if we check further, there was no difference. So just to summarize what I've showed you so far, it seems like the activation of these frontal fields is able to trigger phase reorganization of the beta activity over remote occipital sites, which was the first experiment. This change in the phase, this reorganization in the phase of the beta seems to be perceptually relevant because, as I showed you, there is a change in the performance in the dot detection, the motion detection task, but also is related to the excitability of the visual cortex. And also, it seems like there is some kind of anatomical specificity because the frontal fields seems to influence directly V5, but not so much V1. Not at all V1, actually. It seems that there is a difference. And also, it was a specific effect of the frontal fields that disappeared when we stimulated the vertex, or CZ. So if we take together all these experiments, it seems that there is some converging empirical support that top-down control from the frontal fields is able to control the excitability of the visual cortex by realigning the phase of neuron oscillation that are generated within the visual cortex, and these oscillations are in the beta band. And also, there is some kind of literature that's building up on the beta, so maybe it's in favor of this beta as being a top-down rhythm. And we also thought that because our participants were not doing a task, that maybe this realignment might reflect a general top-down control mechanism. Okay, so I said at the beginning there was this nice paper with monkeys where where the profile of the spikes was the same whether they were activating with brain stimulation or the monkey was actually shifting the attention, and there was a very high similarity. So we wanted to check as well what happens in human. So instead of stimulating the frontal fields, now what we are asking participants is actually to do an eye movement. So we know that saccades are strictly related with, of course, attention, and also that the frontal fields are involved in the generation of these saccades. In this case, there is something a little bit different because there is also a spatial component. So there is something happening only on one side of the visual field. I will explain in a moment. But again, the idea is that this top-down signal or any signal that is generated by the frontal fields, when it's reaching the visual cortex, will phase reset the oscillations, and therefore we can kind of probe the excitability of the visual cortex and be able to reconstruct these ups and downs. So in this case also we use phosphines in darkness participants to make self-paced saccades. So the instruction was simple: whenever you're ready, just saccade to the left or to the right. Try to make it as big as possible, but they were blindfolded. And what happened was that at some point when the saccade was big enough to pass the threshold, the TMS was triggered. And the trigger could happen at any point between 20 milliseconds and 340 milliseconds from the onset of this saccade. And as a control condition, we asked them just to blink. That was very boring, but that's what they were asked to do. And we asked them to report if they were able to perceive phosphines. So we only stimulated one side, the right visual cortex, so they were always perceiving phosphine in the left visual field. This is important for the results. Okay, so these are the results, and you can see here how many phosphines they're perceiving. Sorry. And on the x-axis, you can see the interval from the start of the saccade and the delivery of TMS. This is not as clear, but it seems like there is something there. If we look at the spectral content of their performance, you can see that there is something. So when they do a saccade to the left, which is where they perceive phosphines, there is something in the theta band, but most important for me, there's something in the beta band. When they do saccades to the right, so in the space where they do not perceive phosphines, there is something in the alpha band. And this is what happens when they do blinks. This is their performance in terms of how many phosphines they perceive. But if we look at the spectral content, there was nothing really significant. So when they blink, there's no modulation of the visual cortex excitability. Okay, just to summarize this study, left towards saccades seems to be explained by theta and beta band, right towards saccades alpha band. And in my idea, which is possibly very simplistic, what I thought is that the beta band is processing somehow the relevant information, because we only find it when people are saccading towards the left side, so when they see actually something, where they are processing the phosphine. And on the other side, we have alpha, because when they saccade to the right, they know that there's going to be something on the left, but they tend to ignore it. So they're kind of ignoring what's happening there, and therefore we have alpha. Okay, so now the next question is, okay, we find this phase reset seems to be nice on the visual cortex, but what about another system? So we moved to the motor domain, and in particular, we were interested in understanding if this phase reset could be something that is related to motor inhibition. I guess we all agree that motor inhibition is important because it helps us survive. So for example, if there is a car approaching, you have to stop your action and you should not cross anymore. And there is so the question was really how some of the areas that we know are involved in these motor inhibition, such as the inferior frontal gyrus and the supplementary motor area, how are these areas sending messages to the motor cortex when we have to stop a motor plan? So the aim of the study is to track the effective connectivity between inferior frontal gyrus and plasma of the right hemisphere, I should mention, and just going and check what happens to the primary motor cortex. So I'm not going to explain what motor work potentials are, but the idea is, again, is still the same. So in this case, the sending area is going to be the inferior frontal gyrus on the right, or the supplementary motor cortex. And what we want to know, once again, is whether there is a phase realignment. And therefore, if we go and probe the excitability of the motor cortex, just like the visual cortex, we should be able to see these ups and downs in the excitability of the primary motor area. The task is very, very simple. It's just a go-no-go task. Participants are asked to press when they see a triangle going up and to not do anything when they see a triangle pointing to the right. And there's two conditions. So let's say there is a layer more, there is another layer of complexity here, because after they are presented with the cue, they receive 2 pulses instead of 1. And these might happen very early. And I don't remember when the last one is, but I'll know in a moment. But it's in the early phase of the motor planning. So they receive 2 pulses, and the idea is to go and check whether the communication between these 2 areas and the motor cortex changes over time in a way that is consistent with this phase reset. Okay, so here we started with TMS data. And this is what happens, so this is always the hypothesis, and this is what happens to motor evoked potentials amplitude. You can see the amplitude on the y-axis, and here you can see the time from the cue presentation, so up to 200 milliseconds. And what you can see is that the motor evoked potential amplitude is actually oscillating nicely. It's going up and down, and it doesn't matter if you're stimulating inferior frontal gyrus or the supplementary motor cortex. Sorry. So if we start to look at this performance and we look at what is explaining this data, what we see is that is a nice beta oscillation, so around 20 hertz. And this is what happens when there is a go trial. And as you can see here, there's maybe a small oscillation, but nothing was really significant. So in the no-go trial, there is a nice modulation of the motor evoked potentials, but this doesn't happen in the go trial. We also had some control conditions very quickly. The first one is just stimulating the motor cortex at different intervals just to make sure that there is no modulation just because the motor cortex is involved, and there was no modulation. And then the other question was, it might be that if you're stimulating the SMA, or maybe you're stimulating the inferior frontal gyrus, actually with the coil that is so big, you're still stimulating the motor cortex. So what we tried was also to repeat the go-no-go task, but we also had a condition where we would stimulate the right motor cortex and then the left motor cortex. So we just chose 2 intervals. As you can see in the original condition, which would be blue, in the inferior frontal gyrus, in these 2 intervals that we chose, the amplitude of the motor evoked potential was very different. But if we just look at this condition of the 2 motor cortex, in the no-go trial, there is no difference between intervals. So we took this as evidence that in this case there is no communication from the right to the left motor area. Okay, so we repeated this experiment once more because now we wanted to know what happens in the EEG. So we just chose one interval. Everything is exactly the same. Participants do a go-no-go task, and then they receive at some point a TMS pulse that can be either on the inferior frontal gyrus, right inferior frontal gyrus, on the supplementary motor area. And of course, we are still interested in understanding what's happening on the motor cortex. So these are the results. So this on the left is what we measure from the electrode that is the closest to the stimulated site. So the inferior frontal gyrus is FC2, FCZ for the prisma. And this is the difference between no-go trials and go trials, and this is power. And what you can see is that there is a nice increase around my favorite frequency band, which is again the beta frequency. It's here, but the main point here was the motor cortex. So if we look at C3, which is usually considered to be above the motor cortex, you can see that no matter if you're stimulated inferior frontal gyrus or the SMA, there's still some kind of increase in the beta frequency. This is the topographical distribution of this significant effect. I just want to show you this data in a different way. So you can see the beta power in the go condition for the two stimulated sites and no go. And it's just to say that if we look at the power, there is more beta in the no go compared to the go condition. And one important thing that we also wanted to make sure that it's actually not just C3 that is recording this beta, but it's actually a beta that is generated by the motor cortex. So we just looked at the source of this beta activity, and it is the motor cortex and a little bit of parietal cortex. I'm almost at the end. So just to show you the data exactly the way that I showed the data from the visual cortex. So if we look at the alignment across trials, which is an index of phase reset, we see something that is very, very similar. So here 0 is when, is when the stimulation is applied, and also here we see a significant increase in this intertrial phase coherence in the beta band. And just to finish, one more thing that we wanted to know is whether, if we look at an index of connectivity, which is checking whether there is actually communication, not just an effect on the receiving area, we also see, so this is a phase locking value, just means how synchronized the two areas are over time. What you can see here is that there is an increase over time of the connectivity between the two areas, so inferior frontal gyrus, actually, as you mentioned, and primary motor cortex. But this is only significant for the no-go trials. Okay, some conclusion. It seems like the inferior frontal gyrus and the SMA are involved in changing the excitability of the motor area, but they do something that is looking quite similar. So it might suggest that they work together in movement inhibition in the early phase, actually, of motor inhibition. They do some kind of, maybe they work together. Yeah. Also, the other important thing, just like we saw for the visual cortex, it seems like the inferior frontal gyrus SMA are transmitting information in bursts, so in oscillations rather than just sending one signal or a continuous signal. And also that this network, once again, is working at a specific frequency, which is beta. Just very briefly, general conclusion. So this series of studies seems to support the idea that brain oscillations are involved in communication within neural networks, that this communication occurs in bursts. So areas are not always connected or not connected, but sometimes the influence is going up and down. So the influence on one area over another is rhythmic. And in these experiments, it always seems like the beta oscillations is coming through. So the idea is that this is a rhythm that is heavily involved in top-down interactions within different cortical networks. And I want to thank you for your patience. And I also want to thank you, everyone that was involved in all these studies. Thank you.
Can you hear me? Yes. Thank you, Mima. Are there questions? Okay.
Yes, thank you for the presentation. It was super interesting, super cool data. And I was wondering, have you ever tested whether these phase reset effects are present in peripheral oscillation, motor oscillations during motor behavior, like physiological tremor, some movements, something like that?
Yeah, I think it's a great question, actually, and it would be extremely interesting. I know that there are studies, for example, with tax, where someone has showed that if you start to, I think it's beta frequency as well. So if you set the tax to be in the opposite phase to the tremor, you can cancel the tremor. So there is some indication that is really that there is something to be pursued there. So that would be extremely interesting. Yes, thanks.
Thank you, Mima, for the beautiful talk. I have quite a general question on your first study. So you presented the results about the phase modulation in the beta band, while based on the literature on visual attention, I would expect it to be in the alpha band. So I was wondering, what's your idea? If it's based on maybe most of the studies a priori focus on alpha oscillations, or whether the mechanisms you described are specific for the beta band.
Yeah, okay, that's a very good question, actually. So if you look at the literature, I agree. So most of visual studies are always finding alpha, so alpha is there. But then if you look at them maybe more closely, it also depends a lot on the type of task that you are doing. So in our study, so the one with the dots, everything was presented centrally. And this could be something important. But what I think that will make the difference is whether there is a distractor or there is no distractor. And because in our studies, usually there's no distractor, in these ones at least, I think that there is no alpha that is kind of suppressing a part of the space. And I think, the studies with the saccades kind of support this idea as well. Because the moment that participants have to shift the attention somewhere else, and maybe they have to ignore what's happening on the other side, we see that there is a nice peak in the alpha band. So I think that's why we don't have alpha.
Thank you.
And also why we don't have theta. Because there's also a lot of studies that show that when you look at the oscillations in behavior, for example, there's always this theta that is there, that is everywhere. But then if you look at what they're doing, usually they invite the participant to actually sample the space. So in the moment when you ask people to not just focus on a specific part of the space, but if they have to move around, then maybe there are other frequencies. Thank
you. Hi, very nice talk. I think it's really exciting that you might have a mechanism that we could use to reduce the variability that we see in all sorts of stimulation studies, knowing what area to target potentially using this method. And the only potential downside that I see in using that to, on an individual subject level with some feedback to allow us to, say, adjust the position of the electrodes or some other aspect of what we're doing, is the number of trials or the time that it seemed to take. So am I right that it takes hours in order for you to compute a proper PLV?
Yeah. Yes.
Is there any way that could be done, or you could get a sufficient estimation in a shorter amount of time, I guess is my question, to make it useful in the sense of making adjustments?
Yeah, so actually the TMS EEG study is not that long because I only had 100 trials. And it can be done in a short amount of time. So maybe in 1 hour, you can have something that might be meaningful. So what I would like to do actually is to try and use the behavioral side of things to set intervals that are individually adjusted. That is something I would really like to do, but apparently funding bodies don't like this idea, so they don't give me grants for that. But I think it would be extremely interesting. But the TMS-EEG, I don't think is very long. But of course, then you have to add whatever you want to do afterwards. But I think it's feasible. I think it will be feasible, yeah, definitely.
I like to go back to your frontal eye field experiments, and I find quite intriguing why you found your effect, nice effect, when you look at MT V5.
V5, yeah.
But you don't find effect in V1, and so I wonder if it's due because the type of visual stimulus we are using. You are using moving dots, which is a great stimulus for V5.
Yeah.
But it's not as good for V1.
Yeah.
Because the receptive fields in, well, you know, V5 are huge, the other are small, and so the question is, have you tried, or not, using a different type of stimulus, reversal gratings or something like that, without moving component?
Yeah, that's a very good question, actually. So in the first study, there's a group of participants who's doing the moving dots, and that's not related to phosphines. And then there was another group of participants who was doing the phosphine. In the phosphines experiment, there's no stimulus. So there is no V5 activation generated by the dots. In that case, there was only a stimulation of the frontal eye fields, and then there was a stimulation either of the MT or V1. So in that case, there's no stimulus. We were also surprised to see that there was no modulation of V1. But then if you look at the literature, it's really hard to tell what is connected to the frontal eye fields. And if you look at for example, the attentional literature, what people report is that sometimes top-down control works really well for high order visual areas, but not so well for the primary visual cortex. But there is controversial results because then there's other people who say if you look at the visual evoked potential, even if they're generated in V1, you can still see that there is a modulation if people are paying attention or not. So this is controversial. But it might be, there's also another thing. So there is a study that is TMS combined with MRI, for example, and they stimulate with repetitive TMS the frontal eye fields, and they find a change in activation of V1. And then I thought maybe it's because, we're only giving like one pulse, and maybe because it's very likely that there is no direct tract that is connecting the two areas. So there is a direct connection, as far as I know, between FEF and V5, but there might not be a direct connection between FEF and V1. So maybe the other thing that comes to mind is that one pulse might not be enough to activate the connection between the frontal eye fields and V1. So these are the explanations that I was trying to come up with, but I was definitely surprised.
Thank you.
Thank you, Mima, for very nice data. Going back to your saccade experiment, let's say that you tell the subject that he should expect also phosphine on the right visual field, so ipsilateral of the stimulation. What would be your predictions in terms of results with that kind of instructions?
So if they can perceive phosphines on both sides?
Yeah, exactly. Even in the side where should not actually perceive any phosphines because you're not stimulating the contralateral hemisphere.
I think it depends on what design you would use. So if you're 100% valid trials, then maybe I would say, I would say beta. And then if you have some valid trials and some incongruent rather than invalid. If you might have invalid trials, then in the invalid trials, I would expect that there is alpha coming through, possibly. Yeah, that's my prediction.
Any other questions? Well, I have a last question then. You analyze the data as oscillations, but the phase reset indicates that these oscillations are phase-locked to the stimulus, to the TMS. So what do you think is the relation between your data and TMS evoked potentials? As evoked potentials are phase-locked, do you think there is some kind of like relation between your data and TEPs?
Yeah, so this was one of the questions that we also had in mind, and also the reviewer, actually.
It wasn't me.
It was you. No, the question is, could it be that your data are actually explained by an evoked potential and is not about brain oscillations at all? So we have an analysis, which I actually have somewhere. I have an extra slide if you want to see it. So we calculated how likely it is to be an oscillation. So how much repetitive pattern of oscillations are in the data? Because if it's not significant, then everything we have might have been explained by the evoked potential. So we used a method that is called better oscillation, or BOSC. I should have it, sorry. No, I don't have it. Yeah. Because it's a hidden slide. So basically, this method allows you to reject anything that is not repetitive over time. So you retain everything that is an oscillation, and if it's not there, it's just not there. So it means that it might not be an oscillation. And then we also checked whether the performance of the participant was explained by a model that is not a cosine. I also have it. No. Yeah, so we came up with these alternative models. One was a cosine wave, which is an oscillation, and the other one is an evoked potential. And what we see is that the evoked potential is never better than the cosine wave. So we had some kind of support to the idea that it's not an evoked potential. Yeah.
Okay, thank you. Thank you, Mimma, and thank you all. Now there is the poster session and coffee break. So please go and see the posters, because you remember you are going to vote later.